When it comes to managing blood sugar levels, most people think about counting carbohydrates. However, new research from the University of British Columbia reveals that proteins and fats can also significantly impact insulin production for some individuals. This groundbreaking study, published in Cell Metabolism, is the first large-scale comparison of how different people produce insulin in response to carbohydrates, proteins, and fats. The findings suggest that insulin production is much more dynamic and individualized than previously thought. The research team conducted tests on pancreatic islets from 140 deceased male and female donors across a wide age range. These islets were exposed to each of the 3 macronutrients, carbohydrates, proteins, and fats, while the researchers measured the insulin response alongside 8,000 other proteins. The study's senior author, Dr. James Johnson, a professor of cellular and physiological sciences at UBC, noted, "Glucose is the well-known driver of insulin, but we were surprised to see such high variability, with some individuals showing a strong response to proteins and others to fats, which had never been characterized before." The study revealed that while most donors' islet cells had the strongest insulin response to carbohydrates, approximately 9% responded strongly to proteins. Another 8% of the donor cells were more responsive to fats than any other nutrient, even glucose. This challenges the long-held belief that fats have negligible effects on insulin release in everyone. This research challenges the long-held belief that fats have negligible effects on insulin release in everyone, said first author Dr. Jelena Kolic, a research associate in the Johnson lab at UBC. These findings lay the groundwork for personalized nutrition that could transform how we treat and manage a range of conditions. The study also examined a subset of islet cells from donors who had type 2 diabetes. As expected, these donor cells had a low insulin response to glucose. However, the researchers were surprised to see that their insulin response to proteins remained largely intact. This really bolsters the case that protein-rich diets could have therapeutic benefits for patients with type 2 diabetes and highlights the need for further research into protein-stimulated insulin secretion, said Dr. Kolic. This variability underscores the importance of understanding individual drivers of insulin production for better dietary guidance. With a better understanding of a person's individual drivers of insulin production, tailored dietary guidance could help people better manage their blood sugar and insulin levels. Insulin plays a major role in human health, affecting conditions ranging from diabetes, where it is too low, to obesity, weight gain, and even some forms of cancer, where it is too high. Personalized nutrition could revolutionize how we treat and manage these conditions, offering more effective and individualized dietary recommendations. The research team conducted a comprehensive protein and gene expression analysis on the pancreatic islet cells, providing insights into the molecular and cellular characteristics that shape insulin production. In the future, genetic testing could determine which macronutrients are likely to trigger a person's insulin response. The researchers hope to expand their work into clinical studies that would test insulin responsiveness to the trio of macronutrients in a real-world setting. This research was supported by the Canadian Institutes for Health Research and JDRF Canada. The researchers also expressed gratitude to the organ donors and their families for their invaluable contribution to this study.